All right, so where we left off last time, that we talked about the reasons why cells have to divide eventually, primarily because they become too big. So today we're going to talk in more detail about the actual process of cell division. So again, quick recap: when a cell becomes too large, it copies the genetic information. In other words, it makes a copy of its DNA, and divides in half, each of the new cells getting a copy of the DNA. And in itself consists of two parts or two phases. There's mitosis, which you may have heard of. Mitosis is not the actual division of the cell. All that's happening in mitosis is that we are separating the nuclear material, or if you will, the DNA. All right, we're making sure that the DNA is evenly split, that all the DNA needed goes to one cell, goes to the other. And it's not just a split down the middle, it's every single gene makes its way to each of the two cells. And then the actual division itself is called cytokinesis. This prefix we've seen a couple times this year, cyto in reference to cell, kinesis being some kind of movement. So it's the actual cell, or the cytoplasm itself, is divided, making sure that all the organelles and such are divided evenly as well between the two new cells. So first and foremost, I want you to think of the cell's life as kind of like a cell cycle, or a cycle. And this is a very popular diagram of the cell cycle. If there were a starting point, it would be right about here, because this is where a division occurs, and this is where, if you will, a cell is brand new. It's like a baby. All right, so it basically lasts from one division to the next. So again, this is where the division occurs. It goes through these different phases. It goes through the last process in which it divides again, and then it becomes two brand new baby cells, and they go through their own cell cycle, and so on and so forth. Okay, most of the cell's life is in what's called interphase. So in this graph here, this entire 3/4 of it, give or take, is interphase. All right. If you were to look at a cell under the microscope, and you could tell that it's an interphase because you can see the nucleus. The nucleus is still intact. It's not dividing. It's not separating its DNA. And it's basically doing what normal cells do. It feeds. It grows. If it's a neuron, it's firing off impulses. If it's a blood cell, it's carrying oxygen, and so on and so forth. So it's basically doing its normal cellular functions, but it is growing, as cells do. So the interphase itself is three subphases, noted by the light green, the yellow, and the blue. All right, the G1 phase, which stands for gap, is just following the division here. It's basically when the cell is small and it spends most of its energy growing. Now, because it is growing, because it's getting larger, it's becoming less efficient. So in the next phase, S phase, it starts to replicate its DNA. All right, it's replicating every single one of its DNA bases. The entire chromosomes are being replicated. And then when it enters G2 phase, the second gap, if you will, it's preparing, now that it has a second copy of its DNA, now that it's ultimately large enough, it prepares for the division, which is going to be this last step here. The M phase is that last phase. All right. This last chunk where it looks like there's a lot going on is the M phase. All right, and it follows interphase, and it consists of mitosis, which are these first four phases here: prophase, metaphase, anaphase, and telophase. We'll talk more about. And then it also consists of the actual division of the cell itself, which is cytokinesis, that last little chunk right there. All right, now some background information about the chromosomes themselves. So your DNA does not exist in one long strand. It actually exists in several strands cut into pieces ultimately. That and it's wound around DNA, proteins rather. It kind of keeps it nice and contained and bundled. We as humans contain 46 chromosomes that exist in 23 pairs. We'll talk more about this when we get to genetics. We're not the only species with 46, but one of the notable characteristics of us, genetically speaking, is that we have 46. All right, so this is actually a picture of all 46 chromosomes of a woman, mind you. We'll talk more about the X's and the Y's, but this person has two X chromosomes. They're a woman. So what has to happen before cytokinesis is that each one of these 46 chromosomes has to replicate, all right, and it does. Now, this picture is not as detailed, but the important thing to note is that each of these chromosomes is actually attached to its copy. All right, that's why there's that. It looks like they're tied together temporarily. All right, so what were once single chromosomes are now copies of chromosomes attached, and it's important that they remain attached temporarily. That point of attachment is called a chromatid. All right, it's what holds. Sorry, it's not. It's called the centromere. I apologize. It's the centromere that holds it together. And the chromosomes themselves, another term for a replicated chromosome attached to its copy is called a chromatid. So when we start using that term chromatid, it is in reference to chromosomes, but it's in reference specifically to chromosomes that have copied and are still attached to their copy. So once the chromosomes have been replicated and copied, we can now begin mitosis. So the longest phase of mitosis is prophase. All right. So before we get into it, the best way, I wish I could remember it better, but the 4 steps of mitosis are prophase, metaphase, anaphase, telophase, or what I like to remember, PMAT, P-M-A-T, and the P stands for prophase. It is the longest of the 4 phases. All right. So what happens first is the nucleus begins to dissolve. So this is a cell taken with a special camera under interphase, and as we enter prophase, we can see that nucleus is starting to dissolve. All right, it looks pretty much intact here, but we can start to see it breaking apart. The chromosomes actually start to twist upon themselves and become nice and dense and visible. And these things called centrioles begin to form at opposite ends of the cell. You can see them a little bit here. So they are ultimately going to act as anchors, because this is ultimately going to be kind of like a tug-of-war. And they're actually starting to radiate their spindles, and the spindles attached to the chromosomes themselves. They are physically what's going to pull the chromosomes apart. All right, so there again is interphase, and there it is in prophase. In case you're wondering, this is kind of what the centrioles look like, these little protein bundles, if you will. This is a little artist drawing, so all the chromosomes are kind of floating around. The centrioles exist, they're attached, and they are ultimately attached to the centromere of each of the chromatids. Then we enter metaphase, which is pretty simple in that what happens in metaphase, as you can see in this picture here, is the chromosomes simply line up in the middle. When you look at these under the microscope, it's almost pretty obvious in most cases what's going on here. So you see the nucleus breaking down. Now the nucleus is technically gone, but all that red stuff is the DNA, and it's lining up in the middle. All right, I make the analogy that recess is over, everybody get in line because you're going to be broken up into your different classes. So again, there's prophase rather, and then there's that notable metaphase, which leads us to anaphase. And anaphase, now that they're lined up, they are physically going to be separated. All right, so I make the analogy that they kind of look like spirit fingers of Bring It On fame. So what happens is the centrioles start to pull on the spindles, and they pull apart the attached identical chromatids. So for instance, they were held together in the middle. Now one of the copies is going to go here, one's going to go there, and that's the case for all 46 chromosomes if you're thinking of humans. All right, and then anaphase technically ends when they finally reach the opposite ends of the cell. I know it's hard to see, but this is technically the cell here. And then last but not least, the T stands for telophase. All right, so as we go from anaphase to telophase, you can see kind of a difference there. It's starting to look like 2 cells, and you're starting to see 2 nuclei, because that's pretty much what's happening. Telophase is kind of like the opposite of prophase, too. The chromosomes become less dense. They're starting to loosen up and unbind. The nuclear envelope begins to reform. All right, you're starting to form nuclei again. The spindles are no longer used, so they're broken down. And the cell actually begins to pinch in 2, which is what cytokinesis is, and we'll see that in a minute, leading us to cytokinesis. So basically what we did in mitosis was we separated the DNA so that the cell can actually separate, which is what cytokinesis is. It's the division of the cytoplasm again. Now there is a notable difference between animal cells and plant cells. Perhaps you can think of what that notable difference is that plant cells have to accommodate for a cell wall, whereas animals don't. So animal cells tend to pinch, kind of like a little balloon animal. So we have here early metaphase, anaphase, 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 telophase, telophase, and you can see that pinching. This is cytokinesis down here. Plant cells, though, because they have to build a cell wall between the cells, this is an artist drawing, but you can see here that the cell wall is starting to be built. But here in an actual cell, you can see the interphase. This is actually a really good picture, mind you. This is prophase, metaphase lined up in the middle, anaphase being pulled apart. Telophase is starting to form nuclei. Notice how they are smaller, but you're starting to see, along with the cell membrane, which might be hard to see, but you are starting to see the cell wall being built between the 2 cells.